might suddenly decide to update one of 487,000 updates any second. It's done that before for me. Just start. Shall I go?
Yes. Okay. So, to introduce Professor Andrew Lever, who's at based in Edinburgh School of Clinical Medicine here. This session is to hear about HIV and AIDS in a sort of bigger picture, and then after the talk we'll have some discussion maybe to identify some interesting modelling challenges and modelling approaches to these issues. Andrew, thank you for coming.
Honour, pleasure, terror. So I presume this is a sort of diversionary activity for you really, because there's I don't think there's any numbers in here at all apart from the odd million here and there. And this is based really on a lecture I gave a while ago, which Julia didn't fall asleep during, and obviously felt that was sufficient recommendation to do it again. But I haven't actually looked at this lecture since that time, so it's not going to be too fluent, I don't think. Anyway, I'm an infectious disease physician, and I trained in the late 20th century, which at the time was thought to be a rather inauspicious time to be looking at infectious diseases. And it's thought actually this may be an apocryphal statement, but now it's so well used that everybody likes to think it's true. So William Stewart, the Surgeon General of the USA, said it was all over, and he was not a very good prophet overall, because in the 40 years following that, this is an incomplete list of the emerging infectious diseases that have sought to attack mainly the human race. There's the odd animal agent here, but these are mainly human pathogens. And then there's the re-emerging disease with the drug-resistant TB and bioterrorism, such like. Of all those, the only one that's really been given the epithet of a plague is HIV, which is the causative agent of AIDS. And AIDS was first spotted in this report from the Morbidity and Mortality Weekly report of the CDC in Atlanta in 1981, when it was noted that a small number of young men, all active homosexuals, had been treated in hospital for a very unusual pathogen, usually only associated with people with impaired immune systems, called Pneumocystis carinii. It's now called Pneumocystis jirovecii, but most people remember it as Pneumocystis carinii. And 2 had died, and they already, prior to that, had infection with cytomegalovirus virus infection and Candida. And these are unusual constellations of pathogens for young, otherwise fit people. Those 3 in particular are not the sort of thing you would get if you were fit and healthy. And in the same year, the same journal reported that the same syndrome was appearing in patients who were IV drug users. So the immune system, I apologize if at any stage in this I patronize you a bit by underassuming knowledge, but the immune system is largely divided into 2 big arms. One is the so-called humoral immune system, which is the antibodies, and the other is the cellular immune system, which is the cytotoxic T cells, which kill infected cells and the natural killer cells and such like. And deficiency on one or the other predisposes you to infections with actually a rather specific range of pathogens. So if you're antibody deficient, and about 1 in a million boys in this country is born with X-linked agammaglobulinemia. They make no antibodies whatsoever, but they only get serious problems with this relatively limited range of infections. And some people with antibody deficiency are actually pretty well. So although we make a big fuss, and immunologists in particular make a big fuss about the antibody system and the adaptive immune system, with a few antibiotics you can get away with having no antibodies. And they don't get viral infections in general. If you've got a problem in your cellular immune system, then you get a different panoply of infections, often viruses, intracellular pathogens, and that includes cytomegalovirus, the herpes virus, Pneumocystis, now called jirovecii, and Candida, the fungus. So actually in the 1980s I was working on primary immunodeficiency, people who were born with these sort of problems, and the people who I was working with immediately identified AIDS as something which must have affected the cellular immune system because it predisposed people to that. And so it really wasn't a great surprise when the lab investigations showed that the patients had a deficit in the T helper lymphocyte population, and they're the sort of guiding brain of the immune system, which teach the T cells to kill or teach the B cells to make antibody. The following year, the epidemic, as it was becoming, had spread to another patient population, and those were the group who'd received blood products, particularly patients with haemophilia, who again had pneumocystis, and again had a low level of lymphocytes. And when they were tested in the laboratory, they seemed to have a defect in their cellular immune system. Interestingly, unbeknownst to most people in Haiti over the same period, they had noticed, and this hadn't been reported in the US during that time, that there'd been almost an outbreak of this very unusual cancer called Kaposi sarcoma, originally described by Moritz Kaposi in elderly Jewish men in the Mediterranean region, later identified in Africa, unknown cause. But in Haiti, they'd spotted that this rather unusual tumor was appearing in otherwise young, healthy people, and it forms these purplish patches on the skin and on the mucous membrane, in fact all the way through the gut and the respiratory system. But it wasn't for another 15, 13 years before a virus was identified which caused that, which is yet another herpes virus, human herpes virus type 8, and that seems to be causative of this lesion, but it took a long time for that to be discovered. So things were spreading in the early 1980s. There was a transfusion-related case and then heterosexual transmitted cases, and suddenly it became obvious that it wasn't a disease of minorities or people you could ignore or people who had unfortunate habits or unfortunate medical conditions. Everybody was potentially susceptible. And so a meeting was convened by the WHO in Geneva, because at that time what was now called AIDS had appeared in 12 different countries. In a beautiful piece of zeitgeist, the Soviets said that AIDS was never going to be found in the Eastern Bloc. Again, their prediction capabilities were a bit like Will Stewart's. But as a response, there was the early appearance of paranoia, which accompanies every infectious plague. So blood imports from the USA were banned, and the CDC did their best to try and reassure people, saying that co-workers, friends, relatives didn't seem to be getting this disease, therefore close contact didn't appear to be a problem. Obviously, injections, blood transfusions and sexual contact seem to be the main risks. And then very soon afterwards, I mean remarkably soon in the history of infectious diseases, Françoise Barré-Senoussi at the Pasteur Institute in France isolated a virus from a gay individual who had this syndrome, and showed that in tissue culture it was able to kill these very same lymphocytes which had been shown to be depleted in the patients with HIV. And this is the first electron micrograph picture of HIV, the virus budding from the surface of a cell. Here it is budding off free. She was working with Jean-Luc Montagnier at the time, but the general, this is being streamed live, isn't it? The general opinion is that she was the person who was the virological brains behind it, and he in some ways was a little bit fortunate to share so much of the glory. I'll never be allowed back in France now, I don't think. But okay. A year later, there was this very famous press conference where Robert Gallo, accompanied by the then Secretary of State for Health, Margaret Heckler, announced that the probable cause of AIDS has been found. And he was right, except it wasn't him that found it, because the virus they claimed was theirs was the one they'd actually got from the Montagnier lab to compare with what they were looking at. So he rediscovered the French virus. And Heckler at that meeting famously said, having been told by Gallo that there would be a vaccine for AIDS within 2 years. It's interesting actually reading some of the history as I did preparing this. Margaret Heckler never once in the time she was Secretary of State for Health mentioned the words HIV or AIDS to the then sitting President Ronald Reagan. Not once. So by this time paranoia had sort of gone into full swing, and this is a typical poster that you might have found there, where AIDS is germ warfare by the US government against gays and blacks, and there was this build-up of belief that the virus had been created by the US intelligence agencies, and it was designed to wipe out undesirables in the population. And it went on even further to the point where the drugs which were first discovered actually were quite toxic, and the myth was that HIV didn't cause AIDS, but it was an excuse to poison people who are homosexual with nasty pharmaceuticals. It's quite interesting that the past and present US history of biological germ warfare all substantiate the conclusion. Of course, germ warfare actually started pretty much in the US when, what's his name, in the Indian Wars distributed blankets to the Indians, but only from patients who'd had smallpox. Amherst, that's right, who sadly was British. So the paranoia grew. Ryan White, who became a cause célèbre and a big figure, was a boy with haemophilia. And a majority of parents of other children and a majority of teachers in his school wrote to the head wanting him to be banned from school, and he was banned from school in the US. And he became quite a celebrated figure, friend of Elton John and such like, a huge fundraiser, and illustrated the stupidity of this. And then elsewhere, I think this was Oklahoma, a family called the Kays, who had 3 sons who all had haemophilia, were told to leave, get out of town, and because they didn't, the friends and neighbours burned their house down. And the popular press, notably our own, The Sun, came in AIDS is the wrath of God, says Vicar, and the more right-wing churches in the US in the Bible Belt also joined in. And it's funny in a way to see history repeating itself when a few months ago David Sylvester, the UKIP councillor, said that the storms in Britain were all because David Cameron had allowed gay marriage to be legalised. So some things just don't change, do they? Now, some of you looking around are probably mature enough, should I say, to remember some of the publicity that occurred at that time. And to their credit, they did try, but there were some very obscure things like falling tombstones on the British TV and Don't Die of Ignorance campaigns, which sort of skated round rather a lot and left people rather mystified as to what was going on. But this was the British Reserve response. This was the French-Canadian response, aesthetically perhaps more pleasing and certainly more graphic. And this is the very unhelpful response from UN AIDS. And as I said before, in this position, the very last concern on my mind would be whether I brought my condom. And some of them were just downright awful. This is just dire. I suppose they were trying to make a point very forcefully, but taste just didn't come into it. So around that time, there was this epithet that HIV was affecting a relatively small group of individuals, the gay population, heroin abusers, people from Haiti, and patients with haemophilia. And it was not only small groups who might be marginalized, but it was also a stigmatizing condition. So that some of these conditions that were associated with it, like Penicillium marneffii, which interestingly is a fungal infection you only get in Southeast Asia. You never see this in the West, but it's an AIDS-defining condition down there. And that shows you that the immunodeficiency just makes you prone to the local bug. It doesn't just make you globally prone to every bug, which is the same all over the world. And Kaposi sarcoma, that's a particularly severe case. These are very stigmatizing and disfiguring conditions. And then in one of the most amazing pieces of medical sociology, the 5th H got infected. I guess some of you know who this is. It's Rock Hudson. And Rock Hudson got infected and eventually died. Now Hudson, of course, was a big wheel in the Hollywood community. The Hollywood community were pretty influential with people like Reagan. And there was this sociological turnaround, the like of which I doubt one will ever see again. Because around that time, a little bit afterwards, before this had really taken off, I was at an AIDS conference in Montreal, and shared a taxi back to the airport with a group of pharmaceutical representatives, actually pharmaceutical managers. And they were saying this is a small-scale disease, no one's going to get interested, there's no money in this disease. And they were pretty much writing it off in terms of a financial future in pharmaceuticals. And then what happened here was that the gay population in the US, who were articulate, intelligent, very influential, particularly in politics, turned this whole thing on its head and managed to persuade the establishment that if you were not in favour of eradicating HIV, you were homophobic. And no American politician could possibly afford, particularly on the east and west coast, to be accused of being homophobic. And that really was the tide turning in favour of masses of money and interest going into treating and looking for a treatment for HIV. So where did HIV come from? In the 1980s, the serological tests were becoming more widely available, and it became obvious that although the outbreak had been identified on the east and west coasts here, and it was being found in other countries, if you did serological surveys, Africa, West, Central Africa seemed to have the highest prevalence. And so people who go hunting these sort of things started to look for likely sources of infection in closely related animals. And the one that came up was pan-troglodytes troglodytes, from whom a very closely related virus called simian immunodeficiency virus, and they're abbreviated by the species, so this was CPZ chimpanzee, was isolated not long after HIV itself. And that was so closely related to HIV in sequence that it looked like a very obvious source. When people started looking at other primates in Africa, then a very large proportion of them, it's not quite clear what proportion of them, were found to have a similar virus. So these viruses have been established in primate populations in many different primates in Africa, and this is a sort of family tree of these various isolates. So the question is, how did HIV get into humans? How did SIV get into chimpanzees? And the sequence analysis showed that the chimpanzee virus was a hybrid. It was a chimera of two viruses, one from the red-capped mangabey and the other from the great spot-nosed monkey. And these somehow must have recombined, the two viruses must have mixed and matched. And the likely, or almost certain, mixing pot was the chimpanzee, who had predated both of these. And one must imagine that this chimp, or the chimp, must have predated one of each of these monkeys in pretty close succession, possibly on the same day, for those viruses then to infect and mix and match within that chimpanzee, or certainly in a short space of time, speculation. And then by the same token of a predation course, we acquired it from pan-troglodytes troglodytes through the bushmeat trade, through blood contact, because in the wild these viruses are transmitted by biting and scratching during fighting rather than by sexual contact, and so it's blood transmission all the way to humans. So the original site of transmission was thought to be probably in the Sango Valley up in the Republic of Congo, but later sequence analysis suggested it may have been more likely to be around Kinshasa, and there were lots of sociological events occurring around then during the scramble for Africa, which make it likely that this was the site. And again, looking at sequence data and tracing back, making molecular clocks to go backwards, the estimate is that SIV first transferred from chimps to humans sometime before 1930, but the margins before 1930 are quite wide. HIV-2, a second virus, also transferred across from a different monkey species, the sooty mangabeys in the Ivory Coast, further around here, a little bit later on. And there have probably been 6 or 7 HIV-2 transfers, very few of which, probably 1 or 2 of which have survived in humans, and there have been a number of HIV-1s, but only one has really taken hold and spread. The simple tracking, though, didn't really answer all the questions, because it's clear from molecular analyses of the SIVs in primate species that SIVs have been around for thousands and thousands and thousands of years. So why should it have suddenly happened this late when humans have been in contact with that primate population for many years? And the actual source, Pandrocloditis troglodytes, doesn't actually exist in the Democratic Republic of Congo. A closely related chimpanzee, Schweinfurti, also has SIV, but that virus has never transmitted to humans, and yet there doesn't seem to have been a major difference in contact. SIV-infected sooty mangabees exist in several other countries other than the Ivory Coast, but doesn't ever seem to have transmitted. And as I said, these are transmitted by blood, but not sexual contact. So there's a whole lot of mysteries as to how it happened. And as I mentioned, the scramble for Africa has been invoked as being a major cause for it happening at that time, because of the forced labour that was being imposed on the local populations by the colonising forces, the requirement for more food for these large populations of labour, including increased use of bushmeat, recruitment of a male workforce to urban areas and seaports to actually man these factories and dockyards where all the trading was going on. Wherever you get a male workforce recruited, you get prostitution. And this, interestingly, has been recreated in a number of places more recently, particularly in places like the Middle East, in the Arab world, where, and more particularly in a place like South Africa around the mines, where again you've got large populations of migrant male workers all living in close proximity in communities, and a lot of prostitution going on. And that must have been one of the big contributors to the explosion of HIV in South Africa. Even this doesn't fully account for the fact it spread so fast, and the argument has been that to get the virus to adapt so well to humans such that it could replicate that well requires very rapid serial transmission, so it gets the opportunity to mutate and replicate at high titer. And so one thought has been that the use of unsterile injections, needles, serial use of needles in individuals allows a virus to transmit very rapidly from person to person. And there's no doubt that genital ulceration, which is associated with other sexually transmitted diseases, increases the transmission risk very significantly, so this would have added to this possibility of the virus passaging very quickly. And that's just a picture of Leopoldville as was, and which became Kinshasa in the 1920s. It was quite a cool place, obviously. So there's a lot of evidence for this going over this way to Haiti, people returning to and from homelands, possibly tourism. There's a suggestion that it may have been one single person from Haiti who managed to introduce HIV into the USA. So this is very bottleneck transmission. This is all again based on sequence data. And this was actually taken from a picture from an article in the 1980s, which was called "The Global Spread of HIV." And the arrows that I thought were missing really were these arrows, because after it got into the USA, there was this fantastically huge amplification of HIV from East and West Coast, which has been the driver of the homosexual epidemic. And then, just as a slight aside, Peter Duisberg, in 1987, who was a very well-known scientist, member of the National Academy of Sciences in the US, discoverer of some of the oncogenes, and very highly respected, somehow got, always a bit of an iconoclast, but got this idea that people who had said that HIV was not the cause of AIDS were actually in the right, and the virus was a passenger. It was lifestyle that was causing the immunodeficiency, and because at this time there was very little in the way of treatment and people were dying, a lot of individuals who had the same paranoid theories about the US government making HIV joined in with him. A lot of the uninformed gay population came along and said, this is a good idea, we're being blamed for transmitting a virus which isn't the cause of AIDS to other people. And it's still not clear why he got so obsessed with it, but he did get obsessed with it. And it had remarkable knock-on effects. So in South Africa, not long afterwards, apartheid ended, and after his first term of office, this chap, who doesn't need a label, handed over the control of South Africa to this chap, Thabo Mbeki, who appointed Manto Shabalala Msimang as his Minister of Health. They didn't like the idea that HIV caused AIDS, so they assembled a group of experts, including Peter Duisberg, and you'll see from this Zapira cartoon that these experts include some very famous HIV physicians who, between them, came up with the conclusion that HIV did not cause AIDS. Now you might ask, why would Mbeki want this? Why would Manto want this? And the reason was that there was thought to be a slur being imposed on Southern Africa, whereby the sexual promiscuity, which had been demonstrated very clearly in the outbreak of HIV in the gay population, was being mirrored onto the South Africans. And he didn't believe that there was that level of sexual promiscuity in South Africa. He thought it was an insult. Many Africans thought it was an insult. And they thought they were being branded as really being highly immoral, very badly behaved. And so there was this underlying drive that they wanted to try and prove it wasn't that. And so they went to this extent. There is now a suggestion that this level of sexual promiscuity isn't that high, but that there's a phenomenon called concurrence in South Africa, where one male may have a number of wives on the go at the same time. And I guess it's people like yourselves who've done modelling which suggests that you can explain an equally explosive spread of HIV under those sort of circumstances as you could in the circumstances where homosexual contacts of sort of 20 to 100 in a weekend were occurring to transmit it in San Francisco. This chap Zapiro is the most amazing cartoonist. How he gets away with it in South Africa is remarkable. This was his best cartoon ever. When Manto said that HIV didn't cause AIDS, beetroot, wild African potato, garlic, lemon juice were all going to treat HIV. So he said, all of these vegetables prevent the rollout of antiretrovirals. True or false? False. Lemon is not a vegetable. Again, how he didn't get banged up in prison for that one. It's unbelievable. Apparently he has to leave the country occasionally after he's published a cartoon and can't go. And it's sort of okay to joke, but actually it was awful, because although the prevalence of HIV was increasing anyway before Mbeki came in, after that it just rocketed. And undoubtedly this continued belief or this continued message that HIV didn't cause AIDS, they didn't need antiretrovirals, a rich country which probably could have afforded them even then, allowed the virus really to take hold to the point where something like 20 to 30% of the population on the hoof were HIV positive. And they're still getting over that disaster. There's one interesting other country here which you may or may not have spotted, but while all of these are getting darker, this one here gradually gets lighter, and that's Uganda. And President Museveni lost a son to AIDS, and he took it on himself that HIV was not going to take over his country. And he went on this incredible publicity drive about the risks of catching HIV. It was on every building there were posters about the dangers of catching HIV. If you went to the post office, you got information about HIV. If you tried to get a mortgage, they gave you information about HIV. And he actually managed, simply by that and some obviously decent health practice, but again there were no antiretrovirals right at the beginning here, to halve the prevalence in the antenatal population. So it just shows you can actually do something quite amazing with publicity alone. But this really has been the source of the heterosexual epidemic across the world. And it is international. So we don't have many HIV positive patients in our cohort at Addenbrooke's. We have 200 to 300. But they come from almost 50 different countries. And it just shows the mobility of the virus, because the the virus is as mobile as the people, and the people have become more and more mobile, certainly in the last 50 years. So 30 years on from that discovery, this is where we are, almost up to 30% down here, a very large epidemic occurring over in the eastern part of Europe and across into Russia. China is difficult to judge, but even a very low number, a very low percentage here is still a lot of people. And the areas that I pointed out before. And the actual figures are something like this. The burden is still seriously carried to the greatest extent in sub-Saharan Africa. So this is what the virus looks like. It's a 100 nanometer particle. It's circular. It's got a density under its lipid envelope. It's actually got a conical core, but if it's cut in the wrong sort of section, it looks like it's cylindrical. That's it in cross-section. I'm sorry if this one's going to patronize you, but sometimes some of the people I talk to, particularly ignorant individuals like medical students, don't have an idea about size. And so I say, what's 100 nanometers? I say, what if I enlarged this virus to the size of a tennis ball, and then I enlarged you by the same factor? How big would you be? Anybody want to make a guess? Well, you'd be that size. So viruses are very small, and you can get a lot of viruses in a lot of tennis balls in a place like this. So they're pretty hard to track down. Nonetheless, we know a lot about the structure from a lot of work, and this is the structure of the mature virus. There's that dark layer I mentioned underneath the lipid envelope, the matrix protein. This is the mature virus. There's the conical core, the envelope proteins by which the virus latches onto cells and gets into them, and then inside the two copies of the genomic RNA, each about 10,000 nucleotides long. So again, I'm sorry, this is a patronizing slide, but this is my intro to the life cycle of HIV. So this is a cell, nucleus, cytoplasm, and cells have cell surface receptors, and those cell surface receptors can be bound by external ligands like hormones and such like, and they then commonly will send a signal into the nucleus which may lead to the transcription of a new messenger RNA. That messenger RNA may be trafficked out to the cytoplasm to be translated on ribosomes, and if it's an exported protein like insulin or antibodies, then you can get export of a protein. And effectively all HIV does is take over that same system. So it recognizes the cell that it best replicates in because of its receptors, which it can bind to specifically. They're obviously not there for its benefit, but that's how it gets in. Its RNA genome is copied by this enzyme that it takes in reverse transcriptase to a DNA double strand, and another enzyme that it takes in copies it into the DNA. Viral RNA is produced, viral proteins, and new viruses bud out. So I'll take you through that in a little bit more detail with a comment on the drugs we've got which inhibit that. So here's the first stage. This is the virus binding to its receptors, and there are two receptors, CD4 and a co-receptor, and depending on whether it's on a lymphocyte or a monocyte macrophage cell, it's CCR5 or CXCR4, and it needs both of those to get in. When it binds, there's a fusion event between the lipid envelope and the lipid membrane of the cell, which allows the virus to get in. But we have now drugs, including the archetypal Maraviroc, which are able to bind to CCR5 and actually block the virus from getting in. That obviously will only stop the viruses that use CCR5, but since that's the major transmitted form of HIV, the CCR5 using one, that's very useful. And you might argue, is it not risky sort of putting something on the CCR5 receptor on all of the cells of your body? Hasn't that got a function? And it turns out that about 1% of us in Western Europe are homozygous negative for CCR5. We don't have that protein, we have a truncated version of it, so it's non-functional. And we don't seem to have any ill effects, apart from it seems to be a slight predisposition to a worse outcome with West Nile virus infection, but otherwise we're pretty healthy. And interesting, that mutation doesn't occur in Africa, where you might think it might have evolved in response to HIV, partly because HIV hasn't really been there long enough in humans. When the fusion event occurs, it occurs because the envelope protein, which is a folded up protein, and similar to that in flu, unwinds and then forms a sort of triple coiled coil with a very hydrophobic tip which plunges into the cell membrane. And there's now, well, has been for a while, a drug called T20 given by injection which interferes with that helix or binds to the helix so that you don't get proper entry. The virus has evolved, and some viruses have learned to evade that and actually use the drug to get into cells, which is a rather ironic thing. Once the virus gets in, then there's this reverse transcription step where the reverse transcriptase converts the RNA to DNA. That's the molecular structure of it. We have two groups of drugs, one of which is very large, one is smaller, called the nucleotide reverse transcriptase inhibitors and the non-nucleotide reverse transcriptase inhibitors. When you're building a nucleotide chain, you're putting in adenine, guanine, cytosine, thymidine, etc. And the nucleotide ones are analogues of these, which look like those. So the virus picks it up and puts it in accidentally, but they don't have a tagging on piece, so you can't then carry on the chain. So they're chain terminating drugs. The non-nucleosides are the ones that bind to another part of the enzyme and just make it stick and not work. They're both very powerful drug types. Thereafter, if the virus has escaped that, it's got to get integrated, and in it goes. The integrase enzyme cuts and pastes it into the DNA of the cell. And that, again, is integrase, a viral enzyme, but we now have integrase inhibitors, and these are obviously very powerful drugs which stop the virus getting integrated. Once the virus has got to this stage, it's effectively a cellular gene, so the cell will transcribe it as it will any other cellular gene to produce RNA, and the RNA will be translated like any other RNA. And the RNA will go out onto ribosomes and produce this green protein, the GAG protein, which is trafficked to where the virus is going to bud from. And then the virus has a problem because it's got to get this species of RNA which is being translated is also the species that goes inside the virus particle. So somehow it's got to get some of these molecules down into the virus particle. This is the area we've been working on for God knows how long now. And it turns out that the end of the RNA can fold up into a funny, complicated three-dimensional knot shape, which is distinctive enough for this protein to recognize it. It's called psi, short for packaging signal, abbreviated as the Greek letter psi. And that is recognized by the GAG protein, and this allows it to traffic it to where the virus is budding from. And then two of those assemble into the virus particle, and out they go. And the very last step is the protease, which is again a viral enzyme, converts what was an immature particle to a mature particle by cleaving all the polyproteins. And we have extremely powerful drugs called protease inhibitors, which block that step. So we've got drugs which stop many different parts of the life cycle. There's a big gap here, which is one of the areas we're working on. And we've got some quite promising packaging inhibitors, at least in vitro. Having said that, HIV has been the nonpareil in terms of the number of drugs that have been produced following this sociological revolution, which said we've got to put lots of money into it. And there are now, if you look from 87 to 2013, which is about 25 years, there's about 20 to 30 drugs. There's no other branch of medicine in which that number of drugs have ever been produced in that short space of time, and this is actually incomplete. And they have to be given together. Monotherapy doesn't work. Everyone knows who this is. Do you know his real name? Okay. He's the most famous person ever to come from Zanzibar, Farrokh Bulsara. And he had monotherapy with AZT and then monotherapy with DDI. Had he lived a couple of years longer, he'd have just hit the time when people were giving combination therapy, because a single drug, the virus only has to make one mutation to escape. Multiple drugs, it has to make simultaneous mutations to escape. So the era of highly highly active antiretroviral therapy started about here. Interestingly, one of my friends used to deliver this chap's drugs at night, and because no one was supposed to know about it, he had to disguise himself as a pizza delivery man and turn up on his motorbike at night with his bag of drugs. So what happens with HIV? You get the virus, you get a primary infection, and if you look in the circulation, there's a big rise in virus as it starts replicating. You get the explosive rise in virus in circulation. And at the same time, those lymphocytes which I mentioned earlier on, which get depleted, go right down, telling you the virus is killing them. Then there's a slight readjustment. The immune system starts kicking in and getting a bit of control. The number of virus-producing cells goes down as they're eliminated and they're neutralized by antibody. There's a bit of a recovery of the CD4 lymphocyte count, but overall, the virus wins. It gradually increases. It gradually depletes the immune system. And at this stage, where you've got so few lymphocytes that you can't fight these other infections, you start getting the symptoms and then ultimately the disease AIDS. And that's a reasonable, not sure whether it's a mean, median or a mode, but about 12 years was said to be the average time that it took from first infection to death. With antiretroviral therapy, we inhibit the virus, so it goes down, and the immune system recovers. And it is really quite remarkable. So HIV in the USA went from being the biggest killer, bar none, from nowhere, to coming right down after introduction of HAART. And this is probably going to fall even further, but 5 to 10 per 100,000 is really very trivial now. And this again is how it's represented, that antiretroviral therapy has saved about 3 million years of life. This was your life expectancy at the age of 25 before HAART started, and as it was introduced it got better and better. This is the population control. And then the Danish cohort study, which I think is next. The Danish cohort study was published in 2011. They follow an enormous population of individuals, and they subdivide them into people who have just got HIV, otherwise perfectly well, no other diseases, no other problems with their drugs, and that's group 1. And these people have got other diseases, or they're alcoholic, or they've got cirrhosis. But if you've got nothing else wrong with you, and you take your drugs and you're adherent to them, then your survival curve is not far different, and not significantly different than the normal population. And for a disease which was 100% fatal 20 years ago, this is utterly remarkable. So HIV infections peaked in 1997 globally, and they're gradually going down, and people are living longer. The deaths took a little bit later to peak, but they're going down. But of course, the number of people living with HIV, living healthily with HIV, carries on going up and up. But it is flattening off, and that's an important fact. It is potentially looking quite rosy, but it's far from perfect, and even in the country which is wealthy and which has good access to anti-HIV care, the figures, again from the MMWR, show that this is an estimate of the number of infected Americans, of whom about 90% are actually diagnosed. Only about 725,000 of these are actually linked to HIV care. Less than half are actually retained in care, and less than that are on ART, and even less than that, only about a third of this number, if not less, have got suppressed viral loads. So that's where there's not much of a problem in finance. Paradoxically, because of the enormous charitable outburst and things like PEPFAR, if you look at the percentage of people on HIV therapy worldwide, in some places now it's pretty impressive. 63% of people in sub-Saharan Africa who need it are on it. It's not necessarily easy, you have to be on drug trials, which doesn't make it that accessible for some people, but it's a fantastic change. And just put this in, that HIV infection is unique because you can be well for a long period and still be very infectious. You get no protection against a second infection. It's very well documented you can get second and third infections. I say no one's cleared the virus after infection. There's the famous case of the Berlin patient who had a bone marrow transplant, and the bone marrow came from somebody who had this double negative CCR5, and it seems as though there were no reservoir sites of HIV which have reinfected him, and he seems to have cleared it, but obviously a bone marrow transplant is not something that's accessible to most of the population. And untreated, it's more than 90% fatal. It probably is 100%, but people die of something else before they die of HIV. And there aren't other infections like that. So the future. This is me speculating. I told you about Uganda, and there's no doubt that this is probably the most powerful thing that can be done. And there are good studies in sub-Saharan Africa using sports clubs and such like to try and teach people. There are some wonderful analogies where they're using football as an analogy for risky behaviour, and playing football without a goalkeeper is like not wearing a condom and such like. It's imaginative things like this, and also using cultural issues. One of the successes in Uganda, I think I get this right, is that it was noted that kids got advice from their parents about most things, but they went to their aunts and uncles for sexual advice. And so there was this publicity campaign: if you've got a nephew or a niece, you must be talking to them about anti-HIV. So just as an aside, a year or two ago I was on sabbatical. I spent some time in South Africa in this little area here, in Zitalele Hospital, which is, you can see there's not much in the way of roads or anything. It's very rural. It's quite close to a place which became famous recently, which is Unu, which is where Nelson Mandela was buried. And that's Professor Lever doing his ward round in Africa, seeing his 30-odd very ill patients, making a list of jobs, having no junior staff, going and doing all the jobs, and then coming back to see what had happened to the patients thereafter. And there's me in the AIDS clinic. And this little hospital with about 10 doctors manages to distribute HIV treatment through these mobile clinics to about 2,000 to 3,000 people. Very well. It's unbelievable. And education is very much part of that. In fact, there were some slightly amusing things there where if you start someone anti-HIV therapy, they have to answer questions about what it's doing, what does HIV do. And because everything had to be translated, because my closer never got good enough to do it, I would do this through a translator who was often the person who taught the patient about this. So there'd be a conversation which went, can you ask that person, how does HIV cause AIDS? And she'd do it. And it would come back. He says, I already know, so why am I asking him? Well, tell him I'd like to know, so can you do it? It would come back and said, he says, you know, why don't you tell him? Be quicker than me telling you and you telling him. So anyway, we got there eventually. Why isn't there a vaccine? So vaccines for infections are all successful if people recover spontaneously from it, which says that the immune system can do something by itself. Lots of good vaccines around. But in HIV, there's 60 million human immune systems. None of them have cleared the virus by itself. The best looking one actually was published a couple of years ago, which is a CMV-based vaccine, but that hasn't got any clinical trials yet. And now prophylaxis is the big thing, treatment as prevention, trying to reduce the amount of virus around in everybody so fewer people get infected below the infectious threshold. So what's the infectious threshold of a virus or any pathogen? So it varies from pathogen to pathogen. This is the infectious dose for these. And British Airways, on my trip to Botswana a couple of years ago, fed me 10 to the 5th organisms of salmonella just before I arrived. And I went down like Didier Drogba for my trip to Botswana. It's not known what the infectious dose is of HIV. And this is a very difficult area, because it may not be that they're defective viruses. Most of the viruses are defective. They just actually never get near the cell. But the estimate is you need about 10 viruses to be infected. But they're very, very poor estimates. And there were 2 studies which showed that reducing the amount of HIV around did a lot of good. This one was to see whether treatment of genital herpes reduced HIV transmission. So there are 2 groups of people on treatment for genital herpes, not on treatment for genital herpes. And what they found out, they didn't subdivide them for HIV, so some of them were discordant couples in both sides. And some of them were on treatment and some of them weren't. And the complete bystander result was that those who were on antiretroviral therapy had a massive reduction in transmitting to their partner, and nobody got any conclusions about herpes simplex at all. So this was obvious. And then this was another study looking at a randomization of starting antiretrovirals at early or late, and it was stopped early because, not because of these people, but because their partners were showing a very differential level of infection. So the ones who were on treatment were much less likely to transmit to their partners. So two studies which proved something they weren't designed to prove. And now people are talking about pre-exposure prophylaxis, daily Truvada, but of course there are risks to taking one drug and who would get it. The FDA have now been pressurized into approving it. WHO think it might be considered. Improved therapeutics. There's a lot of drugs coming off patent quite soon, and one pretty good observer of this area, John Bartlett from Johns Hopkins, suggests that even in the US, this disease is going to be treated really very cheaply in a few years as they come off patent. And I'm going to skip through this because this is an irrelevance. The problem with HIV is that some of these viruses which get in go latent. They don't produce RNA. And then there's no protein, so the immune system can't see this cell. There's no enzyme activity, so the drugs don't work. And this latent virus is the final reservoir which no one appears to be able to eradicate, and which means you have to be on lifelong treatment, because if you stop treatment, some of these viruses wake up and repopulate your cells. So we're now part of a large collaborative project between the 5 biomedical research centres to try and wake up latent virus and then therapeutically immunize people to get their immune system to kill the infected cells. So the future model of HIV treatment is that I think people are going to start treatment as early as possible because it saves the immune system from depletion. If we can eradicate latent reservoirs, we will, but it's likely to be like cancer. You do your best, you get it down to a level you can't detect, and you say you're now in remission. And then you go away and you monitor them regularly, and if the virus comes back, you start treating again. So it's a cancer model. But the great advantage over cancer is we've got 30 very effective drugs if the virus does come back. So the outlook is going to be far, far better. I think I'm going to stop there, Julie, because this is a little bit of emotional stuff about health being a human right, which is probably not directly relevant to you. So I'll leave it there.
Thank you. We start with questions and then we'll drift into discussion on issues relevant to
So can I ask a couple of questions? Mostly out of ignorance, really, but also because, well, especially this week, you kind of try to go back to all the things you know and question them and see if you can actually come up with a good answer for them. What's the best piece of evidence that has been found that actually HIV causes AIDS, and that how HIV is transmitted? If from a mathematical point of view, it turns out to be incredibly difficult to work with HIV because of this incredibly long infectious period. So everything that you do now and has an impact in 10 years, it's incredibly difficult when you see the impact to say, oh, it was because 10 years ago that happened. So I still struggle in understanding how people came up with this understanding.
Yeah. It's not a silly question, it's not an ignorant question at all, because actually there are still quite smart, informed people making cogent arguments that HIV doesn't cause AIDS. I've been involved in a legal case recently where a patient garnered all this evidence from everywhere on the planet to categorically prove that HIV didn't cause AIDS, and he'd been poisoned by his doctors for years. And effectively it's all circumstantial. Everybody who's immunodeficient with an AIDS-like disease has got the virus in. Nobody who hasn't got the virus has got a disease like AIDS. People who are in contact with each other where somebody gets the virus from the other person then develop the same syndrome. If you inhibit the virus, then the disease goes into remission. So it's all cause and effect. There are also animal studies where you can infect other animals. You can infect, for example, immunodeficient mice. You can replace their immune system with a human immune system, put the virus in there, and watch the cells being depleted. And you've done it under completely controlled conditions, so you can actually see it does do what it says on the tin in a small animal model. The other concern was that SIV seemed to be in monkey populations for 25,000 years, and they were still going strong. And it's only relatively recently been shown by good DNA studies, actually getting DNA from chimpanzee stool found on the forest floor, that SIV chimpanzee does significantly shorten the life of chimpanzees. So that's not good for chimpanzees. It's slightly reassuring. It's not just us that gets this disease. But it is the overwhelming body of evidence, really, rather than that. Transmission, that again, it was studied very much in discordant couples, in particular in the haemophilic population, where there were, the husband was infected, and then after they were able to calculate how many sexual contacts you might need before the wife got infected, and they knew exactly when the male had been infected because they could trace back to the infected batch of factor 8 or factor 9 that the person had had.
Of course, if I can see husbands in couples with HIV positives and the other partner is negative, not necessarily. I mean, we are looking at couples because we know it's sexually transmitted.
Yeah.
It's a bit of a kind of circular argument. I would probably imagine that maybe there is sexual transmission, but I might just start thinking they spend most of the time together, or it's not necessarily sexually transmitted, or blood transfusion, or maybe as soon as you detect, as soon as you have any means of detecting it, it becomes a lot easier because you can immediately say if somebody got infected or not.
So perhaps the better example is vertical transmission mother to child, where it does happen then, but if you've got another child who was in the house before the mother got infected, they live together with that mother just as much but never get infected. So I suppose it's the comparators of people under internally controlled situations. But they're difficult arguments to refute.
So if you get onto this model you're talking about, HIV treatment with remission and so on, do you think there'll be any political or funding will to do any better? Or do you think that'll be it for, if you get a $200, if you can treat any case, $1 a year and they don't die,
absolutely.
get grandkids,
Yeah.
then you're going to want to push for a better treatment than that, or is that going to be the end of it?
The cheapness is almost the danger, isn't it? That you could end up saying that, why bother, it's like paracetamol. And that's partly the attitude of quite a large number of the gay population now, that they're going to get it anyway and it's treatable, so what's the point in taking precautions? It's almost a relief when I've got it, because I was going to get it. I think people would argue that you're never going to fully eradicate it unless you eradicate it from individuals successfully. I mean, I actually have argued your point that there is no vaccine. In my view, there's unlikely to be another vaccine. There's unlikely to be a conventional vaccine, possibly ever. But that's a very sort of depressed viewpoint, because it's a virus that's completely outrun the immune system. But the numbers worldwide actually are flattening off, and in some countries going down, and that's all by drug treatment. So you could, in theory, say, well, why not just treat everybody forever, and eventually it'll go away. But I think that would be a very, very long tail, and it'd be like polio again. When you only need a few cases, and you stop monitoring, and suddenly there's 12 countries which are polio infected, whereas we thought we were within a hair's breadth of it, eradicating it. The problem is that waking up latent virus is turning out to be very difficult, expensive, and very toxic. And until we get something which, because effectively what you're trying to do is turn on a cellular gene, and if you turn on that gene, you're going to turn on all sorts of other genes in the cell as well. So it's a very difficult target. It's very easy targeting a viral enzyme because there's nothing like that in the cell. But it's very difficult when you're targeting something that the cell does for itself. So we may be being a bit idealistic about it, but there is a drive at the moment to try and eradicate latency as the only way to cure. and partly because of the continuing abject failure of all the millions that have been poured into vaccine studies to produce an effect. But $200 a year is not bad.
It feels slightly mind-blowing to talk about HIV eradication.
Mm.
I just don't see how it can be done in less than centuries without the deactivation.
Yeah. Because reactivation can occur decades after your original infection. And the ones that come up are the ones you can recognize from archival sequences from a long time back. But you might argue also that you do your best to treat other chronic conditions like high blood pressure, but they never go away. But you're still improving the person's quality of life at a relatively cheap cost, and that's the price we pay. So HIV may be accepted as a chronic condition, which an unfortunate group of people have. There are implications in terms of having children and such like. When your virus level is suppressed so low, you are actually very, very poorly infectious. And these days, mother-to-child transmission is almost eradicated in the Western world because treatment at and around the time of birth is fantastically effective. It's far and away the most cost-effective part of anti-HIV therapy. And once you don't get the child infected, you don't have a lifelong of antiretroviral treatment. So that's good.
What's the elephant in the room when you're talking about mass treatment of resistance?
Yeah.
Do you think how far in the future we still have this amazing pipeline of new drugs coming out?
Mm. Yeah, there's resistance to every single drug so far. It's a problem in a small proportion of people who have been on drugs for a long time, because particularly at the beginning a lot of drugs were toxic and people didn't like them, and so they would miss them or they didn't take it seriously, and the message of adherence, because we're not allowed to call it compliance anymore, message of adherence hadn't got through. And where it's a problem is where there are breaks in supply. So in South Africa, where I was, the pharmacy went on strike, and we were sort of that close to running out of one particular drug, which would have meant a couple of thousand people suddenly were on monotherapy. That sort of thing is a real risk. The upside of that in Africa is that they're only using a limited range of drugs, so they're only using about 8 or 9 regularly, so there are backups. And as yet, apart from a very small proportion of people who've been on drugs since the year dot and have got the resistance of one after the other, there's very few people who can't be treated at all. That's another argument for trying to eradicate the virus from them. But there will be people who fail. Actually, the follow-on to this, which is potentially a much bigger problem, is tuberculosis, where the drug resistance is a massive problem, and treatment is just as difficult, and continuity of treatment is just as important. And that's a big pathogen with a very big genome, which can evade anything. And now there is what's called totally drug-resistant TB out there in that area where I was working. So that's, I think, potentially the epidemic that's going to take over from HIV, and is going to be potentially much more dramatic, because also you have to work quite hard to catch HIV. But you don't have to work hard at all to catch TB. You just have to be breathing in the wrong place. So I think the TB epidemic is something which is looming on the horizon, hasn't quite got into the minds of politicians as much as it should have done.
So on that same note, with people living longer and longer with HIV, are there any other, could it potentially open opportunities for other infectious diseases than
If you're living longer, you're healthy, so you're not necessarily predisposed to infection. Once you suppress the virus, then your immune system comes back, and the incidence of opportunistic infection plummets down to very, very low indeed, almost to the normal population level. The incidence of the unusual cancers that are associated with HIV, like lymphomas, goes down very significantly, but not as much. So there's still something like, I think, a 30-fold increase in lymphomas in well-treated HIV people compared to the rest of the population. So infections, I think, if the treatment works, are not an issue. Infection is a problem if it doesn't work, but again, it only then infects people who are immunosuppressed. So for the rest of the population, it's not a problem. Yeah.
So this is definitely going to be a question in the beginning now, so
Great.
bear with me. So as modelers, one of the things we believe, and think this is thanks to a paper that came out a decade ago now, is that something that makes an infection very controllable is how short the duration is between the initial infection time and when symptoms appear, when you can detect the infection.
Yeah.
It strikes me that for HIV, at least in the US, from what I understand, is that period is quite long.
Yeah.
And that might not be the most cutting edge technology, but at least the technology that's used in average clinics, patients are told that if they believe they've had an exposure, they need to wait 6 months to be tested. That seems like a very, very long period of time. So I wondered if there's anything on the horizon to make that shorter, because at least from a modeling perspective, that should help.
You certainly should have a test at 6 months to make sure you haven't got it. You should certainly have a test much earlier than that as well, because there are things which will detect that very early phase where the virus goes up within the first 6 weeks. But a sort of a side issue to what you're saying is that you're right. It's got a very long time before it's easily detected by everybody. It can be detected within the first four weeks very easily. And actually, that's very critical, because that peak in virus in the early weeks is when people are the most infectious. And that's the group that's most difficult to actually target. And that probably is why the argument for the pre-exposure prophylaxis took hold, that if you take your Truvada every day when you're at risk, then you're most likely not to get infected at that early stage where you don't know that you're putting yourself at risk. But that is the problem, and we really don't know how to get to that group of people very, very effectively. In South Africa, it's I'm going to get this wrong. I'm not even going to say it. Anyway, there are more new infections per day than there are deaths. By about 50%.
Because people live longer.
Is that cause and effect? I don't know.
If you put more people on therapy and they live longer, then you would expect deaths to go down.
You would, yeah. I guess so.
I guess we don't know really very much, but do we have any idea how the reactivation works? Why does the latent virus reactivate?
So the genes that the control part of the virus, which says whether it's switched on or not, is very similar to the control part of several of the cells genes, which are involved in that cell dividing. So these are what are called memory cells. These are the T lymphocytes, which say because you caught chickenpox many years ago, you're not going to catch chickenpox again because you've got the memory cells there. You might get shingles when they lose the plot a little bit, but you're not going to get a new fresh infection with chickenpox. And it's the T memory cells which are very long-lived, decades, which seem to get infected. And the model is that HIV is infecting cells promiscuously, indiscriminately, and on some occasions it gets into a T cell just as it's converting into a memory cell and going to sleep, and stops dividing. Therefore, all the factors which are going to trigger the cell to divide are no longer there, and therefore the virus can't replicate. And then when that cell is triggered many years later, because suddenly you go to a chickenpox infected house and that T cell wakes up and starts dividing, then the virus wakes up at the same time. And of course, it doesn't matter what that T cell is aimed at, what antigen it's aimed at, it's got the virus. It doesn't have to be an HIV specific T cell.
So paradoxically, there was no HIV boosting from outside,
Hmm.
latent virus would not reactivate.
If you lived in a antigen-free world, which hasn't been thought of before.
So, to answer a question about what theoreticians should be doing, I'll ask it in a very specific way. Say we are organising a workshop in, 2016, like a 6-month-long thing with lots of modellers. Should we be worrying about HIV? Should we have a week on HIV, or is there nothing useful we can do that isn't being done? Should we be doing TB instead?
Depends if it's a nice venue and you're going to invite me, really. It's an opinion, but I think TB is potentially a bigger issue and a less controllable issue even, because the drug pipeline is really not there. It is better than it was 5-10 years ago, but it's big genome, TB, and it's been with us for a long time, and it knows how to live with us very effectively. So I think that is a big one. You might also have son of SARS coming out and such like any moment. You don't know. That list of emerging diseases may mean that everything else is dwarfed. I don't think HIV will have gone away. There may be some interesting things to look back on in what's happened over the previous decades, and unless a vaccine against my gloomiest expectations has come out, and unless reactivation from latency turns out to be easier than it appears to be, I think we're going to be sort of status quo but slowly moving forwards in a couple of years' time. Then the only things that are changing perhaps are some of the new drugs are turning out to be much more powerful than the old drugs. So the virus is having much more difficulty in developing resistance. And there's a new one out where it looks like as soon as the virus develops resistance to it, it almost cripples the virus. It's an area which the virus has so much trouble evading. So if there's a group of those drugs, then you might then see a definite shift in the epidemiology. And they're very non-toxic modern drugs. Having said that, I needle stuck myself when I was in South Africa and had to go on them for 4 weeks, and I felt I was going to die. It was just awful. So, not advisable.
So, do you interact much with modellers who are modelling that process of the proteins within the cell, or cell-to-cell infection?
Not personally, no. I did at one stage have an idea that it might be interesting to, because viruses like this, in fact HIV and its relatives, are sometimes attenuated, modified and gene swapped around, so they're used as gene therapy vectors. Lentiviral vectors they've been used in the clinic to deliver genes, and I did have an idea that it might be interesting to try and model the efficacy of gene therapy, see what were the spread and the kinetics and the sort of loss of vectors that you got when you injected. You can do it in experimental animals quite easily, and I suppose that might give you a sort of paradigm for in vivo spread of the virus, although they're not exactly the same thing. So I did have a brief dalliance and flirtation with people then, but they didn't get very interested. So Julie's the person who teaches me most maths, but that's all to do with RNA structure, which is again a fascinating field.
So maybe the same question but in a different way. So a lot of modelling has been done for HIV, right? So
Yeah.
But maybe it's a bit of a separate community to the people actually studying maybe HIV in patients and the lab. So I'd be curious to know, even if you haven't done any modelling yourself with people, do you have the impression that some of your insights or some something has been sort of, you realise something about HIV that has been brought to you by models.
Yeah, very much
About
so.
models.
So,
What would these be?
the work that David Ho did a few years ago, looking at the effect of antiretroviral therapy on the decline of virus in the circulation, and then looking at the rate of change of the CD4 lymphocyte count, that was revelatory, because that told us how many viruses were being produced a day, how many cells were being killed a day, and they were orders of magnitude higher than we'd imagined. And that was really done entirely by extremely good modelling. So that was a fantastic insight.
So what you're doing with in-house modelling? I think one more question in sort of using HIV as a way of sort of understanding how other things might emerge in the future. The timeline presented at the start of these 5 guys and then Haiti before that.
Yeah.
But then must have been decades of circulation of something in some parts of Africa. Should there have been some way we would have spotted that?
Well, I guess as I alluded to, the social circumstances in Africa changed quite dramatically in that period of the 19th century, when all of the Western European colonial powers were trying to exploit it, and that's a very plausible reason why, if there was some transmission before then, it hadn't spread very far or very effectively. And again, in small isolated populations, even if somebody did get infected with a virus which was probably less well adapted, they would be more likely to die of something else. Or if they did die, nobody would know what they died of. So there might have been some going on, but it was probably in isolated areas where there was relatively poor documentation. It does look like something happened in late 19th, 30th, 20th century, which made things explode and mushroom, and so it hadn't really been going on to any great extent before then. There's that famous book called The River, which I forget the name. The guy put the whole epidemic down to infected, is it poliovirus vaccine? And that was nicely disproven by my friend, who showed that the archival polio vaccine, which they still had in the freezer, didn't have HIV sequences in it.
No more questions. Thank you very much for coming and talking to us and answering our unexpected questions.
It's all right. I hope it was of vague entertainment to you.